Welcome to Practical Lab 1 of the SQ21403 Fish Health and Disease course. The fish was placed into the distracting tray. If the fish was still alive, it was euthanized before dissection. The external further of the fish were observed. Any abnormality was recorded. If there were abnormalities, samples were collected for observation under microscope or further investigation. This fish was disinfected using 70% ethanol. The fish was dissected to expose its body cavity. The internal organs of the fish, such as liver, spleen, intestine, kidney, and brain, were observed. Those organs was isolated. The tip of the loop was used to touch the infected tissues or organs, which is liver, spleen, or kidney. The media agar lid was lifted slightly to perform the striking. The tip of the inoculating loop was glided back and forth across the agar surface. A new loop was used for the next section of the plate. The previous streak was touched, and streaking was repeated in the next quarter of the plate. Step 4 and 5 were repeated until the plate was fully streaked. The plate was The plate was sealed with paraffin and placed upside down in an incubator for 24 to 72 hours.
A clean, grease-free glass slide was prepared. The center area of the slide was marked using a marker pen. An inoculating loop was prepared and sterilized using a Bunsen burner. If bacteria from a plate were used, a drop of distilled water was added to the marked region using a dropper. The inoculum of bacteria was taken and mixed with distilled water on the slide. If bacteria from a broth culture were used, a drop of the bacterial culture was transferred to the marked slide and the bacteria were spread.
The sample was air dried before the standings. The crystal violet type, iodine 70% ethanoate, and safranines were prepared for standings. The crystal violet type was poured and kept for the 60 seconds, then rinsed with tap water. The samples were stamped with the iodine for 60 seconds, and then washed off with the tap water. The slide was tightened to 45 degrees and a few drops of acetone or alcohol was added and followed by the rinsings with tap water. Safranines was added for the 60 seconds and then washed off with the tap water. the samples were airdropped and observed under a light microscope.
So this is the practical class 3, which is we do the parasite counting. First, we measure the fish's total length and weight to record biometric data. This helps us correlate parasite load with fish size. The next step is weighing each fish individually and recording the data. This information is important for later analysis, especially when comparing parasite load with fish size or weight. Next, we carefully dissect the fish to expose internal organs like the liver, kidneys, and intestines. These are common parasite-infected tissues. And then, we examine the organs visually to identify and count any visible parasites present on or within the tissue. As we dissect the fish, we found our first parasite clearly visible in the tissue. This confirmed that the fish was infected. We then continued by examining another specimen. Upon dissections, we found another parasite present in the tissue. Before beginning the histology, we cleaned the fish surface and sterilized the dissection tools with 70% ethanol. We then carefully dissect the targeted organ, for example, liver or kidney, and immediately transfer it into 10% buffered formalin for fixation. After 24 hours, the sample is transferred to buoyant solution and stored at room temperature to further preserve cellular structure for histological analysis. For the last procedure is tissue preservation for molecular work. First, for molecular work, tool sterilization is more stringent. We clean the dissection set with 70% ethanol, soak in 99% ethanol, and flame it using a Bunsen burner. We then allow it to cool. We dissect the target organ under sterile condition and place the tissue into 70% ethanol. The sample is then stored in the refrigerator or freezer until DNA extraction. At the end of the procedure, we store all the samples and label them so we can easily identify each one during future analysis.